AP Bio Review Part 1, brought to you by my AP Trade. Water is the most abundant molecule in living organisms and possesses a variety of instrumental properties that result from its hydrogen bonding and polar bonds. It is the universal solvent. The pH of the aqueous environment inside a cell and its organelles influence many biological activities such as the shape of proteins, proton gradients across membranes, and the speed at which enzymes catalyze reactions. An acidic solution contains more hydrogen than hydroxide. A basic solution contains more hydroxide than hydrogen. The following are the properties of water that make it an important molecule. Cohesion, which is the ability of water molecules to stick together. Adhesion, which is the ability of water to adhere to other molecules. Heat of fusion, which is difficulty in breaking the surface of water and create surface tension. High specific heat capacity, which means it heats up and cools down slowly. Heat of vaporization, which means water's high specific heat prevents it from evaporating easily. Monomers are building blocks of larger macromolecules called polymers. Condensation reactions combine monomers by removing water. Hydrolysis reactions break down polymers into their monomers with the addition of water. There are four macromolecules: carbohydrates, proteins, lipids, and nucleic acids. Carbohydrates consist of monosaccharides, disaccharides, and polysaccharides. The most important monosaccharide is glucose, containing 6 carbons, 12 hydrogens, and 6 oxygens. Lipids are water-soluble molecules composed of glycerol and fatty acids. Fats, or triglycerides, are energy storage molecules consisting of one glycerol molecule with 3 fatty acid molecules attached. Saturated fatty acids do not contain a double bond and are more likely to be a solid at room temperature. Think saturated equals single. Unsaturated to contain a double bond and are more likely to be a liquid at room temperature. Phospholipids consist of one glycerol molecule with 2 fatty acid molecules attached as well as a polar component. They can self-assemble into a bilayer arrangement that is the basis of all biological membranes. Wax and steroids, including cholesterol, are lipids with more complex structures that have a variety of functions. Proteins are polymers made up of different combinations of 20 commonly occurring amino acid monomers. Proteins have a wide variety of functions, including structural components of cells and tissues, transport in the cell's membranes, and the ability to speed up chemical reactions in the form of enzymes. Amino acids share the monomers of proteins. Amino acids undergo dehydration synthesis to form peptide bonds. When they come together, during bonding, there is always a central carbon atom covalently bonded to 4 atoms or functional groups. One of the 4 is always a hydrogen atom, a carboxyl functional group, and an amine functional group. The 4th component is a variable R group, which is different for each amino acid. Proteins have 4 levels of physical structure. Primary structure refers to the specific linear sequence of amino acids in a polypeptide. Secondary structure is the initial folding patterns of certain lengths of the polypeptide chain, such as alpha helices and beta pleated sheets. Tertiary structure refers to the overall shape in which a polypeptide eventually folds. Quaternary structure arises from the association of 2 or more folded polypeptides to form a multi-subunit protein. Nucleic acids are made from monomers called nucleotides. A nucleotide has 3 parts: 1 5-carbon sugar, a phosphate group, and nitrogenous bases. The 5-carbon sugar is either deoxyribose as in DNA or ribose as in RNA. In DNA, you have adenine, thymine, cytosine, and guanine as the nitrogenous bases. In RNA, you have adenine, uracil, cytosine, and guanine as the nitrogenous bases. Nucleic acids have ends, defined by the 3' and 5' carbons of the nucleotide's sugar. The direction in which other nucleotides are added to the chain during DNA synthesis and the direction in which transcription occurs are both determined by the placement of these 2 nucleotides' ends. Enzymes are proteins that act as catalysts to speed up biochemical reactions. The function of enzymes is to lower the activation energy of a reaction. The activation energy of a reaction is the energy required to initiate a chemical reaction. The enzyme combines with the substrate or molecule that the enzyme will act upon. The shape of the enzyme's reactive site matches the shape of the substrate molecule. When the enzyme and substrate are joined, a catalytic reaction takes place, forming a product. The enzyme can be recycled and used for later reactions. Enzymes are affected by pH, temperature, and substrate concentration. Enzymes function at an optimal temperature and pH. As the substrate's concentration increases, the speed at which the reaction occurs increases up to a maximum level at which all enzyme molecules are processing substrate molecules as fast as possible. Cofactors and coenzymes can also affect enzyme function. Sometimes the interaction between them causes a structural change, and therefore the enzyme's activity rate changes. Let's move on to cells. All cells are either prokaryotic or eukaryotic. Prokaryotic cells are simpler and more ancient than eukaryotic cells. Prokaryotes have no nucleus, only a nucleoid region with 1 circular DNA, no membrane-bound organelles, cell wall. Eukaryotic cells are those of protists, fungi, plants, and animals. They contain a nucleus, a nuclear envelope to protect DNA, nuclear pores to allow transport into and out of the nucleus, linear DNA, and membrane-bound organelles. Plants have chloroplasts, cell walls, and a vacuole that is absent in most animal cells. Both prokaryotes and eukaryotes contain a plasma membrane, cytoplasm, and ribosomes where protein synthesis occurs. A few examples: organelles are mitochondria, which is the powerhouse of cell, produces ATP, and site of aerobic respiration. Nucleus, which is site of chromosome storage and RNA synthesis. Rough endoplasmic reticulum, which is site of protein and membrane synthesis, including detoxification and drugs. Golgi apparatus, which modifies and transports proteins for export from the cell. Ribosomes, which are the site of protein synthesis. Lysosomes, which are site of phagocytosis or digestion. Vacuoles, which are storage locations in plants and provide turgor pressure for plant support. Chloroplasts, which are site of photosynthesis. Cytoskeleton, which is a network of tubules and filaments that controls cell shape and causes movement of chromosomes and organelles within the cell. Cilia and flagella are hair-like projectiles that contribute to movement. A plasma membrane separates internal environment from external environment and allows substances to be transported in and out of the cell. Membranes are selectively permeable, meaning that it allows some substances to pass through it, but not others. Selective permeability is because of the membrane structure and captured in the fluid's mosaic model. The membrane is a mosaic of proteins that are embedded and are attached to the phospholipids. The lipid portion of the membrane is composed mainly of phospholipids. Phospholipids have a hydrophobic or water-fearing tail and a hydrophilic or water-loving head. The cytosol and the fluid outside the cell are both aqueous watery environments, forcing phospholipids to form a bilayer. Cholesterol is also found in membranes and helps to keep the membrane fluid. Embedded proteins in the cell membrane can be hydrophilic or hydrophobic. The two types of proteins found in membranes are integral and peripheral. Integral proteins are transmembrane proteins with hydrophobic and hydrophilic portions. Peripheral proteins bind to integral proteins on the outside of the cell membrane. Functions of membrane proteins include transport, enzymatic activity, signal transduction, and cell communication. Passive transport and active transports are the two main types of transportation. Passive transport does not require the cell to use ATP. Diffusion is a type of passive transport in which a substance moves down its concentration gradient from an area of higher concentration to an area of lower concentration. In facilitated diffusion, transport proteins move molecules down concentration gradient. Osmosis is also passive transport and causes water to move across the membrane from a hypotonic solution to a hypertonic solution. A hypotonic solution has lower concentration of solute or dissolved substances than a hypertonic solution. If two solutions have equal concentrations of solutes, they're isotonic. Equilibrium is no net movement of water across the plasma membrane. Active transport requires the cell to use ATP to move substance up the concentration gradient. Specific membrane proteins are used in active transport. The sodium-potassium pump is an example of a protein that uses active transport to move ions through the cell's membrane. Exocytosis and endocytosis are also active transport and move large molecules. Exocytosis is the fusion of vesicles and molecules with the plasma membrane to release stuff to the outside of cell. Endocytosis is when the cell takes in the molecules via vesicles that fuse with the plasma membrane. Pinocytosis is the uptake of liquids. Phagocytosis is the uptake of solids. Now let's move on to discussing energy and metabolism on a cellular level. Energy is the capacity to do work. First law of thermodynamics states energy can neither be created nor destroyed but can change from one form to another and be transferred. An example of this is photosynthesis, where plants take light energy to make chemical energy. Second law of thermodynamics states every energy transfer increases entropy or disorder of the universe. Free energy is energy available in a system to do work. Organisms need this free energy to grow. Exergonic reactions release free energy. Energy is found in the product side of the chemical equation for exergonic reactions. In catabolic reactions, reactants are broken down to produce products containing less energy. The energy released can be used for reactions that require energy. Endergonic reactions require free energy. In anabolic reactions, reactants are joined together to produce products containing more energy. Adenosine triphosphate carries energy in its high-energy phosphate bonds. ATP is formed from adenosine diphosphate and inorganic phosphate. Conversely, when ATP is broken down into ADP and P via hydrolysis, it is an exergonic reaction because energy is released that can be used in endergonic reactions. In addition, ATP can donate one of its phosphate groups to a molecule, such as a substrate or a protein, to energize it or cause it to change its shape. Living systems require consistent input of free energy and an ordered system. Energy input into the system must exceed the loss of free energy in order to maintain order and to power cellular processes. Metabolism is the totality of all chemical reactions that occur within an organism. Cellular respiration makes ATP for cell use, uses glucose and oxygen, makes waste products of carbon dioxide and water, occurs in mitochondria, and NADH is the electron carrier used. First type of cellular respiration is glycolysis. Glycolysis occurs in the cytoplasm and is anaerobic. It rearranges the bonds in glucose molecules, releasing free energy to form ATP from ADP through substrate-level phosphorylation, resulting in the production of pyruvate. Another type of cellular respiration is the Krebs cycle, which occurs in the mitochondrial matrix and is also called the citric acid cycle. Krebs cycle occurs twice per molecule of glucose. Pyruvate is oxidized further, and carbon dioxide is released. ATP is synthesized from ADP and inorganic phosphate via substrate-level phosphorylation, and electrons are captured by coenzymes. The coenzymes carry electrons to the electron transport chain. Another type of cellular respiration is the electron transport chain and chemiosmosis, which occurs in mitochondrial matrix. The electron transport chain captures electrons, pumping hydrogen ions into the intermembrane space of the mitochondria. Electrons are accepted by oxygen molecule forming water. Concentration of hydrogen builds up within intermembrane space, lowering the pH, and ions rush through ATP synthesis into the mitochondria matrix. The rush of ions spins ATP synthesis protein, causing ADP and phosphate to join, forming ATP by oxidative phosphorylation. The chemical equation for photosynthesis is reversed from cellular respiration. The products become the reactants and the reactants become the products. Photosynthesis has two main steps: light-dependent reactions, which is the absorption of light energy and its conversion to the chemical energy of ATP and the reducing power of NADPH. Light-independent reactions, which is the use of ATP and NADPH to convert carbon dioxide to sugars using the Calvin cycle. In light-dependent reactions, photophosphorylation occurs. Chlorophyll and proteins are embedded in the internal membranes of chloroplasts in thylakoids of the grana. They pass electrons through an electron transport chain. When electrons are passed, they allow hydrogen ions or protons across the thylakoid membrane. The formation of the proton gradient powers the process of ATP synthesis. Water is used, and oxygen is released as byproduct. The energy converted from sun into chemical energy of ATP is used to build sugar in Calvin cycle. In light-independent reactions or the Calvin cycle, carbon fixation occurs in stroma of chloroplasts. ATP and NADPH generated by light-dependent reactions are used to assemble glucose. Anaerobic fermentation is when there's no oxygen. In this scenario, the cell only goes through glycolysis followed by fermentation. Fermentation recycles NAD needed to restart glycolysis. Examples of anaerobic fermentation is alcohol fermentation in yeast cells. Another form of anaerobic fermentation is lactic acid fermentation, which occurs in muscle cells when overworked. Thanks for watching. Please don't forget to like, comment, and subscribe.